Hello and welcome everyone to the next session in the GenScript webinar series. I'm Keshava Santavada, Marketing Specialist for Protein Services at GenScript. And I'm here to kick off our webinar today by introducing the presenter, Hongxing Yu. Hongxing is a senior scientist in the GenScript protein production group. She got a PhD in biology from Friedrich Schiller University, Jena, in Germany. With specialization in medical biology. Hongxing has extensive experience in protein interactions between hosts and pathogens using different expression systems. The topic for today's webinar is efficient, stable cell pool cell line development for large-scale antibody and protein production. As many of you would know, two expression vector systems dominate the antibody production in mammalian cell culture. One is based on Dihydrofolate Reductase or DHFR genes, and the other on Glutamine Synthetase genes, the GS system. And in this webinar, Hongxing is going to review some basic information about generating a stable cell pool, cell line, using the Glutamine Synthetase system. She'll also compare the usability of the GS system alongside the DHFR system. And Hongxing is also going to discuss how to efficiently screen stable clones for increased expression and how to monitor the consistency, reliability, robustness, and potency of products during scale up. To ensure the sound quality of this webinar, all attendees have been muted. To ask questions, just type your question in the chat box and Hongxing will address them at the end of the session. I also want to let you know in advance that a quick poll will show up at the end of the webinar, and it would be great if you could take a moment to provide feedback about today's session. And so with that, without any further ado, I'll hand over to our presenter. Welcome, Hongxing.
Okay, thank you for introduction, Kashav. Ladies and gentlemen, welcome to my session. Today, I would like to introduce you this efficient method for high productive stable cell line or cell pool generation, which is popularly used for large-scale antibody production in biopharmaceutical companies. The following topics will be discussed today. Firstly, I would give a brief overview of the stable cell line generation, and then I would like to introduce you our GenScript CHO-GS system, including the principles, development, capability, as well as some case studies. So firstly, let's have a look how is a stable cell line generated. The basic workflow for stable cell line generation includes expression vector construction, transfection into the host cells, select the positively expressing clone under the selection pressure, and positive clone expression, feedback culture, and then going to the cell line generation and process development. So normally the therapeutic antibodies are mainly produced in the mammalian host cells, and currently the CHO cells are the major workhorse for the production of the recombinant antibody. And CHO cells do have some advantages, and they easily adapt to the production medium and grow to a very high cell density and yield the stable glycosylation of the antibody. So the popular CHO cells lineages include the DUXB11, DG44, and the CHO-GS cells. DUXB11 and DG44 cells don't have the dihydrofolate reductase and so-called the DHFR activity, so which makes them suitable for DHFR-based selection system, while the CHO-GS cells have the endogenous DHFR activity. So another cell line is the NS0 myeloma cells, and these cells are also used for the antibody production because they are non-immunoglobulin secreting cells, and they also lack of the endogenous glutamine synthetase, so-called GS enzyme activity, which makes them suitable for the GS selection system. So I will give more introduction later for both systems. As to the expression vector, the mammalian expression vector typically contain two cassettes, and one cassette for the antibody gene and selectable marker genes for expression in the mammalian cells, and the second cassette for the genes enabling plasmid replication in bacteria. So we could imagine that replication origin is very crucial characteristic for any plasmid. The popular used origins are SV40 origin, F1 origin, and the PBR 322 origin, which you could see in this map example, these three origins. And they are used for different expression systems. To achieve high levels of antibody expression, normally a strong promoter like the CMV promoter and the EGF1 alpha promoters are used to drive the antibody heavy chain or light chain expression. Oftenly, an intron sequence in the 5-terminal untranslated region is also included after the promoter to increase the export of the transcribed mRNA from the nucleus to the cytoplasm. And one or more 3-terminal polyadenylation signal sequences are also included for maximize the mRNA level. So in addition to the transcription, the translation and secretion of antibodies are also required. Typically, a Kanzak sequence is placed immediately in front of the first translation codon to enhance the translation initiation, while a signal peptide sequence is placed immediately in front of the mature antibody peptide to direct the antibody secretion. So after the transfection with the certain expression vectors into the host cells, the transfected cells will be further selected by different selectable markers. And these markers can be further categorized into two groups, the metabolic selectable markers and the antibiotic selectable markers. But for CHO cell-based stable cell line, the popular used is the metabolic selector markers. We use the methotrexate, so-called MTX, for the DHFR-based selection system, and we use methionine, sulfoxamide, so-called MSX, for the GS-based selection system. And both reagents, the MTX and MSX, the concentration can also be increased stepwise to further increase the cell protein production by further gene amplification. So there are several reasons which contribute to the expression level. Firstly, the integration of foreign genes is very randomly. The integration itself could induce the disruption of the genome by gene amplification systems. As a result, so the cell clones obtained during the cell line development are highly heterogeneous. And furthermore, high-producing clones are typically rare in a population of transfected cells because the active region supporting this high gene expression in the chromosome is rare. And these high producer cell clones typically have lower growth rate because a significant portion of the resources are being used for expression of those recombinant proteins or antibodies. So therefore, the screening of a large number of cell clones is commonly required to isolate the high-producing clones. So far, the single-cell cloning or serial limiting dilution is used to ensure that the selected cells are producing the target recombinant antibody. And then the additional steps of cultivating the cells and the protein or antibody titer measured by ELISA are used to determine the protein or antibody productivity of the clones chosen for the further expansion. So after the right cell clone was picked, feeding a good medium is also very important for the single clone because it affects the process performance. As we know, most amino acids making up the primary structure of the protein or antibody is directly from the culture medium. So there are also several methods to find the efficient feeding strategy. The component titration is the classic approach, and it involves performing a series experiments to determine the dose response to different media components by adding each of them into the individual cultures. So the media blending is a method that rapidly generates many new media by simply mixture of different media. And spent medium analysis is to measure how a media getting to be changed during the culture process. And automated screening mostly focus on the throughput screening. So that's our feeding strategy. So now I guess we already have some concepts on how to generate an efficient stable cell line for antibody production. So now I would like to further introduce you how did the GenScript generate our CHO-GS system. As I mentioned previously, two expression vector systems dominate antibody production in mammalian cell culture, one based on the DHFR system, selection system, and the other one based on the GS selection system. The GS selection system is based on the glutamine synthesis. As you know, the glutamine is an essential amino acid, and many mammalian cells require exogenous glutamine for cell survival. Generally, the glutamine could be synthesized from the glutamate on ammo by the glutamine synthetase, so-called GS enzyme. So in the absence of the glutamine in the growth medium, this GS enzyme is essential for cell survival. You could imagine that. But this can be further inhibited by the MSX, which could act as the glutamate analog and bind to the GS. So normally the CHO cells, they express sufficient endogenous GS to survive without the exogenous glutamine. But the endogenous GS could be inhibited by the MSX, like I mentioned before. And when an expression vector encoding the gene, the target antibody gene, and the GS gene, when this such kind of expression vector were transfected into the CHO cells, it allows the continuously glutamine synthesis. Only the transfectants with additional GS activity could survive, and this is the principle for the GS selection systems. The transfected gene amplification could be then further induced by the stepwise MSX concentration. So the GS technology was originally developed by Celtech Biologics in 1987. So during the year from 1987 to 1995, there were several patents filed. And then in 1996, Lonza required the GS technology with the acquisition of Celtech Biologics. From time on, the GS technology got to be widely accepted for biologics production. So far, over 100 companies and academic institutions use this technology, and hundreds of the high-yielding cell lines generated with several biologics in clinical trials. And our company, GenScript, started developing the in-house CHO-GS cell lines in 2014, since the majority of the existing patents expired. So in the beginning of this year, we launched our CHO-GS-based stable cell line and cell pools for guaranteed gram-level recombinant antibody production. So as you already know, achieving high antibody production is a combination of many factors, as I mentioned before, including the cell line, expression vectors, screening, feeding strategy, and the overall process development. And GenScript, our objective is to develop a mammalian expression platform combining all these factors that would lead to a rapid and efficient and high producing cell line. So what did we do? We de novo synthesized expression vector with minor modification. As for the host cell line, we purchased the original clone from the ATCC and did a lot of optimization work on the original clone. And currently, we have well-optimized suspension CHO-GS single clones in serum-free medium. And then we conducted a lot of internal validation and developed case studies before launching this service at the end of April this year. So here is, I show you the map of our vector developed in-house. And the gene encoding your target antibody can be codon optimized. And you have a strong promoter that drives the antibody gene expression and a relatively weak promoter to drive the GS gene expression, and coupled with the selection using the MSX, which selected for the integration at transcription level. And also our GS system facilitates rapid creation of high productivity cell lines. As I mentioned before, that the cell line was derived from the ATCC clone. After that, we go through several rounds of the clone optimization and screen of over hundreds of clones. And finally, we optimize the suspension CHO-GS clone in the serum-free medium. So here I show you again the cell line development workflow. Start from the codon optimization, gene synthesis, and construct design, and followed by transfection and stable cell pool screening and evaluation. And next comes the preliminary clone evaluation and expansion. And then you choose the target cells going for further clone evaluation and expansion. So, and then we go into the feedback mode. Typically, we can achieve gram per liter expression during the first feedback round, and then we can achieve even higher during the second round. At this point, we can back the cell or conduct the further evaluation or process development. So now let's have a look what we could achieve using our GS system. So although there are several advantages of using our CHO-GS system, but I would mention the three most important attributes: the productivity, the speed, and the stability. So the high manufacturing cost for recombinant products, especially for the therapeutic products, means that maximizing the productivity is very essential for the establishment of the economic process. So use of the cell line could reliably result in high-yielding cell line. So with the maximum expression levels attainable depend on the product itself, but we can create cell lines producing greater than 5 gram per liter recombinant antibody with the production rates in the range of 5 to 100 PCD. So you can see here that growing from the pool to the feedback one, we can already achieve over gram level per liter expression. And going into the second round feedback, we can take it beyond 5 gram per liter expression level. The speed at the development stage of any biotechnology project is also quite crucial. So the CHO-GS system allows for rapid selection of the high yield cell lines during the first screen. So you could even get higher yields through the media and process optimization, and the significantly reduced time required for generate a cell line is suitable for the GMP process. So gram level recombinant antibody production could be achieved in 14 to 17 weeks, as shown in this graph. So here is also a timeline comparison between the GS system and our DHFR system. So the workflow is nearly identical, except for the fact that you could easily knock off 10 to 12 weeks from the GS workflow. And of course, in the manufacturing run, it might make sense to have the MSX amplification to induce the selective pressure and improve yields even further. But speed here we mentioned is very crucial. So 10 to 12 weeks knock off you could achieve. So the GS cell lines then are consistent in terms of product concentration, growth, and product characteristics can be also easily created. So as you can see here, cell lines maintained over a period of 40 generations demonstrated the consistent product titer and viable cell densities. You could see they are quite consistent. Here I would like to show you one case study of our single cell clone screening process. And this is showing the productivity in the 96 well plates. And the purple bar represents the number of single cell clones with their respective titers shown on the x-axis. As you can see, the titers can go up to 300 milligram per liter at this stage. And those clones with high titers were further selected for the batch culture. So, and here is the batch culture evaluation. We can already hitting close to a gram per liter expression in the batch culture. So again, the clones with the highest titer were further selected for the first round feedback culture. So in the feedback culture round 1, as you can see from this result, most clones exceed gram per liter expression level at this stage, and with antibody titers as high as 3 gram per liter. And from this point, 25 clones with the highest titers were chosen for the second round feedback culture. And after the second round feedback culture, the expression level could achieve 5 to 6 gram per liter. So this is our endpoint. And this is another case study. Here shows the growth profile of a single clone expressing antibody in a feedback model using the Schick flask, and after 2 rounds of the process optimization. And you could see here the peak viable cell density reached 6 million cells per milliliter during the round 1 feedback, and then go up to 10 million cells per milliliter during the second round. And the cells get happier with the feeding and the process optimization. So that was about the case study. Coming to the characterization capability of your recombinant antibody or protein, we have the following capabilities, like the SDS, the LCMS, the SEC, HPLC, and so on, the endotoxin testing. Those methods can ensure that your recombinant products adhere to the highest standards as you expected. So finally, to summarize, our CHO-GS cell line uses a very robust expression vector technology and selection via the glutamine metabolism to provide rapid development of high-yielding and stable mammalian cell lines. And the process is highly scalable. Our cell lines are CHO-derived and adapted to suspension growth in animal-free and medium and the chemically defined medium. And they have been selected for superior growth and expression. And using this tool, we have developed a fully optimized cell line development process that can give you very high production with a high probability of the monoclonality. So that was my presentation about the stable cell line service capabilities. I hope you found it useful. And now I will quickly hand over to Keshav so I can look through the questions you have been typing in. And thank you for your attention.
Thank you very much, Hongxing. I really enjoyed your presentation. I see that we have questions coming in. But before we go there, I just wanted to take a few minutes to tell our audience a little bit about GenScript. Headquartered in Piscataway, New Jersey, GenScript is the world's leading biology research service company. We are internationally recognized as a leading contract research organization, specializing in biological research and early phase drug discovery services. We provide services and products to scientists in 86 countries around the world. And we have built the best-in-class capacity and capability for biological research services encompassing gene synthesis and molecular biology, peptide synthesis, custom antibodies, protein expression, antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now, there are more than 13,000 peer-reviewed journal articles that cite GenScript's services and products, making GenScript the most frequently cited biology contract research organization in the world. I hope you'll take some time to visit our website and check out the many ways in which we can help accelerate your research. So with that, let's again go back to Hongxing, who will address your questions. Over to you, Hongxing.
Hey, Keshav. I'm looking for the questions, but I don't know if there is any technical problems here. I couldn't see any.
Okay.
So
let me see if on our end I can figure it out. Okay, Hongxing, one of the questions that comes up is, are we using this technology to produce proteins and recombinant antibodies, or is it just for recombinant antibody production?
Okay, so we produce both for protein and for antibody. And for antibody production, we can offer some guaranteed service. But for protein production, we need to analyze the protein properties and then to decide if we can guarantee or not. So we have two different kinds of service.
I see. Okay. And could you tell our attendees, give us an idea of how many CHO-GS cell lines have you produced so far?
Okay, so the CHO-GS cell lines was launched, as I mentioned before, the April of this year. So from that on, we developed around 15 cell lines.
Okay. Great. So also curious, an attendee listening in will be wondering, what the end deliverables are. What do you give the customer when they place an order and you deliver it?
Sorry, Keshav, I didn't really get that.
So what are the materials that are provided to the customer? What are the end deliverables?
Okay, the end deliverables. So we can deliver the single clone and with the primary cell bag, and but we don't give our vector information. So basically, we can give you a final report with the cell bag and give you the information for the characteristics of the cell lines and some certifications as well. Yeah, the only confidential things is our vector information.
Okay. So one question that came up is what can cause cell line instability?
Oh, okay. So for this question, if you really have the single cell clone, I could guarantee that there is no instability. But for cell pool, it has a kind of risks because the cell pool is not very pure, and so normally we offer the over 80 or 90% of the positive clones in the cell pools, but still the integration itself could induce some, for example, the random mutations and genetic drifts for the CHO cell, for the host cells, and this can also exhibit the genetic changes. So this is one reason for the instability, and the other reason is the non-transfected cells. They normally grow faster than the transfected cells because they don't need the resource for the expressing antibodies, so they just grow, and then the stable pool could get lower purity and so on. This is also another reason for the instability. So I would say these two reasons, but no worry for the stable cell line.
I see. Great. Thanks, Angshan. That was useful. So I know you addressed this in your presentation, but another question that came up is what media we use for cell culture work.
So for cell culture work, at the moment we use the commercial cell culture, so medium.
Okay.
And those medium, the components of the medium you can easily find from the website, and you can download them. So it's just a commercial medium, but we are also working on developing our own medium.
Okay. And you said for the CHO-GS cell line, you use animal chemically defined medium, right? Protein-free
yes.
serum. Yeah. Okay.
Yes.
Okay. And so one more question is, do we carry out developability studies for the recombinant antibody?
Yes. We. You mean the stability assay, or you mean the characteristic of the cell bank?
So I understand developability in general for the antibody.
The stability. So we normally do the stability assay. So during different generations of the antibody, and to test if they have the consistent producing yield or like that. So this is our basic procedure to before for QC and before delivering the stable cell line. Is this the answer for your question? So I'm afraid that I missed some point.
partly, but yeah, it some degree addresses the issue. Great. I think there's one more question. They're asking about the feed system. Do we have a feed system? Can you tell a little bit more?
Which system? Sorry.
The feed.
The feed system.
Yeah.
Oh yes. Yes, we do the feed system. You mean how do we generate the new feeding medium, right? So we also doing this. We have different components by hand, and we testing them. Yes, so we generate some recipes for the specific feeding medium, but to keeping the consistent result, at the moment we just use the commercial one. So But our other departments, they are trying to have our production catalog products for that.
Okay. Okay.
Mm-hmm.
Great. So yeah, I don't see any other questions at this time. So thank you very much, Hanshing, for answering all the questions. So before we end the session, I wanted to inform our audience about the upcoming webinars in our series. So Hanshing, if you could go over to the next slide, please.
Okay.
Thank you. So I wanted to inform the audience about the webinars in our series that are shown on this slide here. I also wanted to remind everyone that the slides and recording of today's webinar will be archived on our website. I encourage you to visit the website link shown on this slide on the top in a few days and take advantage of these information-packed resources. And if you have any questions that come up later, certainly feel free to contact either me or Hanshing using the email IDs shown up here on the slide. As I said at the beginning, it would be great if you could take a moment to complete a quick poll right after the session ends to tell us how you like the presentation. Your feedback will also help us to improve our services. So again, thank you all for your time and kind attention. And on behalf of the entire team at GenScript, we wish you all success in your research. Thank you very much.